Good day everyone, today we'll be discussing DNA technology. So recombinant DNA technology is the use of in vitro molecular techniques that manipulate fragments of DNA to produce new arrangements. So in the 1970s, first successes in making recombinant DNA molecules were accomplished by the Stanford University. So gene cloning, it refers to the process of making many copies of a gene. So laboratory methods necessary to clone a gene were devised during the 1970s, and then after that many technical advances have enabled a gene cloning to become a widely used procedure. So what are the some uses of gene cloning? First, we have the DNA sequencing. So clone genes provide enough DNA subject the gene to DNA sequencing. So the sequence of the gene can reveal the gene's promoter, regulatory sequences, and coding sequence. So it is also important in the identification of alleles that cause cancer and inherited human disorders. We also have site-directed mutagenesis. So it can be manipulated to change its DNA sequence. So mutations with gene help to identify gene sequences such as the promoter and regulatory elements. So studying mutant gene can also help to elucidate its normal function and how its expression may affect the rules of other genes. So mutations in the coding sequence may reveal which amino acids are important for a protein structure and function. So labeled DNA strands can be used as probes for identifying similar or identical genes or RNA, so it can also be used to localize genes within intact chromosomes. So expressions of cloned genes. First research, it can help elucidate its cellular function. So the gene can be placed next to an active promoter and then introduced into a culture of cells that will express a large amount of protein. Then this aids in the purification of large amounts of proteins that may be needed for biochemical and biophysical studies. For biotechnology, it can be used to introduce bacteria to make pharmaceutical products such as insulin. So it can also be used in plants and animals to make a transgenic species with desirable traits. So in the clinical trials, clone genes have been used in clinical trials including involving gene therapy. So vector comes from the Latin meaning carrier. So a small DNA molecule that can replicate independently of host cells, produce many identical copies of an inserted gene. So purpose is to act as carrier of the DNA segment that is to be cloned. So the vectors commonly used in gene cloning were derived originally from two sources. We have the plasmids or viruses. So plasmids, they are small circular pieces of DNA. They are found naturally in many strains of bacteria and occasionally in eukaryotic cells. So many occurring plasmids called R factors carry genes that confer resistance to antibiotics. So plasmids, they have inside bacterial DNA in the plasmids. So there are commercially available plasmids and they are genetically engineered for effective use in cloning experiments. So those commercially available plasmids contain unique sites where geneticists can easily insert pieces of DNA. Viruses are the alternate type of vector, so they infect living cells and propagate themselves by taking control of the host cell. So it can be used as vectors to carry other pieces of DNA. So some vectors used in cloning experiments, we have P. blue strip. It is a plasmid and used to clone small segments of DNA to propagate them in the E. coli. We also have YEP24 or the YEP24. It is also a plasmid and an example of a shuttle vector which can replicate into two different host species, E. coli and Saccharomyces cerevisiae. So it carries the origins of replication for both species. We also have GT11. It is a viral vector derived from bacteriophage. So the fragments of the DNA are inserted next to the promoter for the lac operon DNA is expressed in E. coli. So it is an example of an expression vector. It is used to clone coding sequence of genes so that they are transcribed and translated correctly. So the SV24, another virus vector, they naturally infect mammalian cells. So they are used for cloning and expressions of genes in mammalian cells that are grown in the laboratory. We also have baculovirus, a virus that infects insect cells. So it can be grown in liquid media, often expressing large amounts of proteins that are encoded by a clone. So PCR or the polymerase chain reaction, it is very common nowadays. Why? Because the testing or the confirmatory test for those with COVID-19 ang gamit ay PCR. So PCR, polymerase chain reaction, it was developed by Carey-Mullis in 1985 and used to make large amounts of DNA in defined region that is flanked by two primers. So there are three steps. We have the denaturation, primer annealing, and primer extension. So palatandaan lang namin dyan dati is DAE. So denaturation, so separate DNA strands with high temperature. So the temperature may be 94 to 95 degrees Celsius. Then primary annealing, lower temperature naman 50 to 56, allows primers to bind to template DNA. And then the primer extension, it is incubated at slightly high temperature, 72 degrees, to allow DNA synthesis to occur. So please remember all the temperatures included for each step in the PCR. So terminologies, primers, they are oligonucleotides, short fragments of DNA, usually 15 to 20 nucleotides in length. Amplification, many copies of the region have been made. So primers, they are the key reagents in the PCR experiment. They are not made in a research or clinical laboratory, but instead at a university or industrial facility. Template DNA is a sample of DNA that contains a gene of interest. So DNTPs or deoxyribonucleoside triphosphates. So it is a thermostable form of DNA polymerase such as Taq polymerase or D thermus aquaticus. It is necessary because PCR involves the heating steps that inactivate most of other natural forms of DNA polymerase. So the starting material is a complex mixture of DNA. So the two primers bind to specific site in the DNA because their bases are complementary at these sites. So the end results, the two region flanked by two primers containing the gene of interest is amplified or yung pinaparami. In other words, the two regions between two primers have been cloned. So first denaturation, 94 degrees for 1 minute, then annealing, lowering of the temperature, then elongation ay at 72 degrees Celsius. So yung double-stranded DNA, i-denature mo yon. So ibig sabihin maghihiwalay. Tapos yung annealing stage, meron kang primer. Yun ay sa 5' at 3' na position. Tapos extending or yung elongation. So it is carried out in a machine known as thermocycler that automates the timing of each cycle. So experimenter mixes the DNA sample, the DNTPs, and the Taq polymerase and excess amount of primers together in a single tube. So it is placed in the thermocycler and the machine sets to operate within a defined temperature range and the number of cells. So during each cycle, the thermocycler increases the temperature to denature the DNA strands, then lowers to allow annealing and extension. So it typically have so the cycle lasts for 2 to 3 minutes and then repeated. Then a typical PCR run is likely to involve 20 to 30 cycles of replication. So PCR technique can amplify the amount of DNA by staggering amount. So assuming 100% efficiency, intervening region between two primers increase approximately 1,000,000-fold. So An important advantage of PCR is that it can amplify a region of DNA from a very complex mixture of template DNA. So it can be used to amplify a sample of chromosomal DNA non-specifically. So non-specific DNA amplification is used to increase the total amount of DNA in very small samples. So reverse transcriptase PCR. So PCR is also used to detect and quantitate the amount of specific RNAs in the living cell. So RNA is isolated from a sample and mixed with reverse transcriptase. So it is a primer that binds near the 3' end of the RNA of interest. So this generates single-stranded complementary DNA, which can then be used as a template DNA in a conventional PCR reaction. So class ito yung ginagawa sa COVID-19. Kasi ang coronavirus is an RNA, so para matest siya sa PCR, ang ginagawa is the reverse transcriptase PCR. So the end result, the RNA has been amplified to produce many copies of DNA. So this can detect the expression of small amounts of RNA from a single cell. Real-time PCR, it allows researcher to follow the amount of a specific PCR product in real time. So they are derived from template DNA that was initially added to the reaction. This allows the researchers to determine how much DNA, such as DNA that encodes a specific gene, was originally in the sample before PCR was conducted. So the carried out in a thermocycler that has the capacity to measure changes in the level of the fluorescence that is emitted from the probe that are added to the PCR mixture. So fluorescence given off by the probe depends on the amount of the PCR product. So PCR allows quick identification of genetic markers of the bacteria, virus, and paternity testing. So it requires only small amount of DNA and can repetitively the DNA synthesis reaction. So PCR in medicine, PCR has had a major impact on prenatal diagnosis of single gene disorder, important in carrier testing for genetic diseases, improves speed, accuracy, and technical flexibility over previous methods. In prenatal diagnosis, it is used to amplify from fetal cells obtained from the amniotic fluid. So many diseases can be detected such as Tay-Sachs disease, phenylketonuria, cystic fibrosis, hemophilia, Huntington's disease, Duchenne muscular dystrophy. So PCR is also used to detect HIV. So allows direct detection of HIV genomes in patients' blood before appearance of HIV antibodies. So high risk of cross-contaminating samples with small amounts of amplified DNA from the previous samples requires extra precautions to prevent false positive. So diagnosis of middle ear infection known as otitis media, and also for Lyme disease. So Lyme disease is a painful joint inflammation caused by bacteria transmitted by tick bites. So most sensitive and most specific tests for the Helicobacter pylori. So it can also detect 3 STDs. We have the herpes, papillomaviruses, and chlamydia. In forensics, PCR can generate sufficient DNA from a single cell, so results are obtainable within 24 hours.